Good morning. Good morning. Am I audible or not?
Audible, sir.
I said good morning, boss.
Good morning.
Well, in the previous class, we were discussing regarding the disinfectants. We have seen what are these disinfectants and what is their probable mechanism of action. Because the different disinfectants they act by various mechanism, and also we have seen the classification of the disinfectants based on their chemical nature. So when we say disinfectants, so these are the chemical agents or the biocidal agents which are used to kill or inhibit the growth of the microorganisms. Probably these chemical agents, so they kill almost any type of the microorganisms. The different chemicals they kill different organisms, but except for the spores, so where only few chemical agents they are having the ability to destroy or or kill the spores, the bacterial spores, which are highly resistant. So apart from that one, the different chemicals they act by different mechanism. And now we have to see or we have to discuss regarding the factors which will influence the activity of these disinfectants. Because the disinfectant they act by various mechanism, and you can see this disinfectant they are nothing but the chemical agents, and this disinfectant activity is nothing but a chemical reaction. We can see here the before going into the factors, before discussing the actual factors which will decide the disinfectant property of or disinfectant activity of these chemicals. So you know that the some of the chemicals like such as the alcohols, biguanides, and chlorine-based compounds, phenols, so they act on the cell membrane of the microorganisms, especially by increasing the permeability of the cell membrane, changing the potential, or even the inhibiting the ATP synthesis, because we know that in bacteria the ATP is synthesized in the cell membrane, and also it will alter the this electron transport chain. And the chemicals such as the aldehydes, biguanides, peroxygens, and phenols, so they may directly act on the genetic material, that is either DNA or sometimes the RNA, by inhibiting the replication of DNA or altering the structure of the DNA. And alcohols, biguanides, peroxygens, and phenols, they also act as a biocidal agents. They induce the biocides autocidal activity, that is by accumulation of the free radicals in the organism. And these free radicals, they bring about the various chemical reactions, the fastened chemical reactions, thereby inhibiting or by altering the metabolic activity of the microorganisms. And some of the chemicals such as the aldehydes and chlorine-based compounds, iodophores, especially the halogens, and peroxygens and phenols, they also inhibit or they react with the thiol, amino, and sulfityl groups, and thereby altering the metabolism of the microorganisms. And pyroxygens, specifically, they target the ribosomes, and they destroy or alter the structure of the ribosome, and thereby bringing about these protein synthesis and other things will be inhibited. And alcohols, they denature the proteins. We have studied that. And some bisphenols, they act upon certain enzymatic activity in the lipid biosynthesis. And this lipid biosynthesis is inhibited, and thereby the cell membrane formation of the cell membrane will be inhibited, and thereby killing the bacteria or the microorganisms. And also these alcohols, they bring about the cell lysis. Alcohols and also the phenols, they bring about the cell lysis, and some surface active agents also. So they bring the pores, the formation of the pores in the cell wall of the bacteria, and thereby killing the bacteria. Aldehydes, chlorine-based compounds, and phenols, once again, they act upon the cell wall by adsorption. And the aldehydes, biguanides, and phenols, so they also act on the coagulation of the cytosols. So some of the proteins and cytosols are coagulated, and thereby inhibiting or bringing the changes in the metabolic activity of the organisms. So if you see this, these chemicals, or what we call them as the biocidal agents, so they're having the toxic effect on these microorganisms by various mechanisms on the different parts of the organism. Some on the cell wall, some on the cell membrane, some on the genetic material, on the enzymatic activities, specifically on certain particular enzymes, and inhibition of certain metabolic activities, specific metabolic activities. So if you see this, the reaction which is involved here, so it is between two different chemicals. So one chemical is a biocidal agent, another one is the chemicals which are present in the as a part of the microorganisms. And this is a pure chemical reaction. And since it's a pure chemical reaction, so whatever may be the mechanism, this chemical reaction, sometimes it may be physical or sometimes it may be chemical reaction, all are affected by certain factors. This reaction is affected by certain factors. So there are a number of factors which will influence the activity of these disinfectants. So some of the factors, they increase the activity, certain factors, they reduce the activity. So in order to understand the activity of this disinfectant, it is very, very important that we need to understand these factors which will affect the disinfectant activity. So before starting that one, so you please keep a notebook and pen with you, because there are certain calculations are involved here. You may have to do that one. So if you don't have one, if you are sitting with just with a mobile, please arrange for a piece of paper and the pen. Is it clear?
Yes, sir.
Yes, sir.
Okay, so there are, as I told you, there are a number of factors which will affect the disinfectant activity by various different mechanisms. So of them, the most important factors today we are going to discuss are, the first one is the time of contact. So as I told you, it's a chemical reaction. So definitely the time duration, they are the time at which the chemical agent is in contact with the microorganism is very, very important. And as we can expect, so definitely the increase in the time of contact, definitely it increases the activity. And the second one is the concentration of the disinfectant. So definitely, since it's a chemical reaction, the concentration is a major factor which will affect this disinfectant activity. And certainly when we have what we have studied in the chemistry, as per that one, so as the concentration of the disinfectant is increased, so definitely the activity of the disinfectant activity will the microbial activity will also increase. And third one is the temperature. So this is another important factor which is a cofactor with the first two factors, that is the time of contact and concentration of the disinfectant, and along with the temperature. So the change in the temperature, so that will change it will affect the time of contact, the required time of contact or the required concentration. So usually, since it's a chemical reaction, the increase in the temperature, always it will enhance the chemical reaction. We know that. And similarly here also, with increase in the temperature, the activity of the disinfectant will also increase. And apart from that one, so these are the three major factors, especially the time of contact, concentration of disinfectant, and temperature. And all the three, they are interrelated. By increasing the concentration, you can reduce the time of contact. By increasing the time of contact, you can reduce the concentration. And by increasing the temperature, you can reduce both the time of contact as well as the concentration. So these first three factors, they are very important, and they will be derived, and we are going to understand certain mathematical derivation. And with that one, we will be able to calculate the actual activity of these disinfectants. And apart from that one, the next factors will be the type of organism present, its number, and the condition. So definitely we know that all the organisms, they do not react similarly with certain disinfectants. Or on the other side, the one disinfectant will not act on the all the microorganisms. So definitely the change in the initial bioburden, what we call it as the bioburden. So initial number of the organisms and their condition and what type of organisms are present. So based on that one, so the disinfectant activity will be considered, or it will be affecting. And next is the presence of organic matter. So this presence of organic matter, once again, is a matter of concern, especially in the hospitals when we are doing the disinfection of a place, so which will be soiled with the blood, pus, and other materials. So this presence of organic matter, most of the time it reduces the activity of certain disinfectant. We are going to study that. So which are those disinfectant which will get affected by the presence of the organic matter. And next one is the hydrogen ion concentration. So definitely the hydrogen ion concentration, that indicate that's nothing but the pH of the formulation, or pH, so which will affect. So the different disinfectant, they act in the different pH level. Some disinfectant, they are very good in the acidic pH, and some will be very good in the alkaline pH. So that's how this hydrogen ion concentration affect the disinfectant activity. We'll study that one. And then comes the surface tension and its role in the formulation of the disinfectant. So definitely when we do the formulation, we use certain surface active agents, and how this concentration of the surface active agents, or what we call them as the surfactants, anionic or the non-ionic surfactants. So how they affect the formulation, how they increase the activity of the formulation, and how it will be, when it will be reduced. So that we are going to discuss in that particular part. And next, the chemical structure of the disinfectant. Naturally, so with the alteration of any disinfectant, the alteration of the chemical structure of the disinfectant, sometimes it may enhance the activity, sometimes it may reduce the activity. By taking example of the phenol, we are going to discuss, so when the activity will be increased and when the activity will be decreased, and what type of changes can be made onto these disinfectants. And nature of the surface to be disinfected. So which surface we are disinfecting, whether we are disinfecting the floor, we are disinfecting a wall or a glass, or what is the type of the material which we are disinfecting. So that also is one of the factor which will affect this disinfectant activity. And finally, the potentiation, synergism, and antagonism. So here, by mixing or by adding the one disinfectant to the another disinfectant, or by combining two antimicrobial agents, so how the effect takes place, or by combining certain chemicals on with this disinfectant, whether the activity will be reduced. So whether the activity is potentized, or it will show the synergistic effect, or sometimes the mixing may show the antagonism effect. So when it will happen and how it will happen, so that we are going to discuss. So these are the various factors which will affect the disinfectant action. And this part is very important, and most of the time you may expect this question in a long essay or in a short essay form. And it is asked in the short essay form, definitely the effect of the time, concentration, temperature may be asked, and in the long essay, so all the factors you will have to explain. So be careful. So this is the most important part in the disinfectants chapter. So now let's begin with the first one, that is the time of contact. So if you plot a graph of a viable count of the bacterial population against the time. So when a population is subjected to a lethal environment, so that is lethal environment is by exposing it to the disinfectant. So it will give a death or the mortality curve. So that means, so when you expose material or substance to this disinfectant material, and the concentration of the population, bacterial population, so when it is exposed, so then definitely as the time reduces, so the number of survivors will also reduce. So this is what we call it as the death or mortality curve. So this then mortality curve, it was studied by the Madsen and Neyman in the year 1907, and by Chick in the year 1908. So they worked independently, and they have showed that the course of disinfection to approximate that the unimolecular reaction. That means, as I have told you earlier, this is just like a unimolecular reaction. It's just a simple chemical reaction. So A plus B is equal to C. So similarly, here the chemical reaction will take place, and this follows the principle of first order kinetics, and it can be applied for this disinfection process. I hope you people have studied this first order kinetics, the chemical reaction in +2 or maybe in the first year of Form D in the chemistry. Am I right? Do you know?
Yes, sir.
Okay, so first order kinetics of the reaction. So as per that, if you apply the formula, K is equal to 1 by T log B, capital B divided by small b. So you just please write these equations, because in the future, in the next, you may have to do certain calculations. So for that one, you may need these equations. Write it down in a piece of paper, and then later on you can make the fair note afterwards. So here in this formula, that is K is equal to 1 by T log B by B. So K, it is a measure of efficiency of the disinfection. So that means, so it's a constant, and it measures the efficiency of the disinfection. So that will be given by 1 by T, that is time of viable count to fall from capital B to B. So where capital B is the initial number of the organism, that is, so initially how many number of organisms are present, and the B is the final number of organisms after the time T. So that means here we are going to expose a certain number of the microorganisms, if you take it as a capital B, with certain disinfectant for a period of T time, that T, and the number of organisms will be reduced to that small b. So this will give the efficiency of the disinfectant activities. Those that will be given by the K. And as I told you, this graph, that is the time of contact, the death curve. So there are three types of the death curves are obtained in the disinfection process. So we don't always get this straight line here. So the very first curve will be the curve A. So if you plot a graph of time versus log of survivors, that is the log percentage of the survivors. That means as the time goes, so when you expose a group of microorganisms or a lot of microorganisms to the disinfectant for a particular period of time, so not all the organisms will be killed simultaneously. You know that we have already studied that one. And initially the number of organisms, initially the sensitive organisms will be killed, and then the other organisms, and finally the most resistant organisms will be killed. So that's why, so after a particular time, when you plot a graph of time versus the log of survivors, you will get usually a straight line. So, and this is the first line. So what we got, that is the line A. So this is a disinfection process which will obey the first order kinetics. So first order kinetics, that is equal to K is equal to 1 by T log B by B. So that kinetics, when it is exactly followed, so you will get this particular graph. But this graph will be obtained only at a particular, only few times, but not every time. We don't get this type of the curve, that is a straight line is not obtained. In fact, most commonly obtained graph will be a sigmoid curve. So most of the time what we get is the sigmoid curve, and this sigmoid curve you can see, so it is divided into three stages. In the first stage, there will be a slow initial kill. So that usually mainly some sensitive and susceptible microorganisms will be killed, and the number of organisms which are getting killed will be very less. So that is the first stage. And in the second stage, there will be near linear rate of the kill, similar to the first order kinetics for the average resistant organism. So that is the part B. So in the part B, you can very clearly see that it is a straight line. And this is almost a straight line, and it follows the first order kinetics in the second part. And then the third one is the slower death rate of the resistant organisms. So that is what we see here is the C. So this sigmoid curve, it can be divided into three parts, that is A, B, and C. So A, the first part, it will be usually a slow initial kill of the mainly the sensitive and susceptible microorganisms. And then the B, which is a near linear rate of kill, similar to the first order kinetics, where the average resistant organisms will be killed. And at the C, so in the later stage, usually the most resistant organisms will be killed, and that will take place. So that's what we get the C. So this is what happens most commonly when you expose a material for the disinfectant activity. And when we get this C, so this C curve is obtained with the higher concentration of the disinfectant. For the same disinfectant, if you increase the concentration, either double or fourfolds, when you increase, then you will get this type of the curve, that is the C curve. So usually this will be very fast disinfectant process. Usually in the initially beginning, it will be very fast, and then later on, because of some resistant organisms, the rate will become a little bit slow. So this is how we are going to get the death curve. So three death curves will be obtained usually. So one is a first order which follows the first order kinetics, that is A, which will give a straight line, but usually occasionally this curve will be obtained. But most commonly obtained curve is a sigmoid curve, which is divided into three parts, that is A, B, and C. A is a slow killing in the initial beginning, and then a near linear killing of the average resistant organisms, and the resistant organisms will be killed in the C part in the later stage. And at the same time, if you increase the concentration by multiple folds, so then you will get a graph C. So is this clear?
Yes, sir.
If anybody has got any question, you can ask now itself, because I know this is a mathematical expression. It could have been better explained with the chalk and talk. So please, if you have any doubt, any question here, you can stop me anytime and you can ask your question.
Explain once again, sir.
Well, see, the death curve, it is nothing but when you expose a group of microorganisms to the disinfectant. So this disinfectant, they are nothing but the chemicals, and the disinfectant activity is nothing but a chemical reaction. Because these chemicals, the disinfectant or the biocidal agents, they react with certain chemicals which are present in the microorganisms. It could be a cell wall, cell membrane, DNA, or any protein or the enzymes. So that reaction, it follows a first order kinetics. That is what you study in the chemistry. So that first order kinetics is nothing but the K is equal to 1 by T log B by B. So that capital B is the initial number of organisms, and small b is the final number of organisms after a particular time of T. So definitely the time plays a measure also. It will take certain time. Not all the organisms will be killed simultaneously at once when you expose them to the disinfectants. So some organisms will be killed in the beginning, and then the other organisms and the resistant organisms will be killed in the later stage. So if you plot a graph of log of percentage of the survivors on the y-axis and time on the x-axis, so definitely you're going to get a straight line. Okay, so this is the straight line. But this straight line which we get the first, that is the A, it is very rarely it is occurs, because it is a theoretical straight line. But in commonly, so when you expose this disinfectant for this the disinfectant activity, so what you get, the graph will be a sigmoid curve. That is what you see the graph, that is the graph B. So this in the B, it can be further, it can be divided into three parts. So initially there will be a very slow kill. The number of organisms killed will be very less in the A part. So where usually the more sensitive and susceptible organisms will be killed in the beginning. And then in the B part, it is almost near linear, or what we can call it as a which follows the first order kinetics. So because after a certain time, the chemical reaction will be enhanced, and the organisms will be killed. So in this portion, so most of the moderately resistant organisms will be killed. And later, the highly resistant organisms will be killed in the later stage, that is the C. So that's why you will get a sigmoid curve. So that's why you will get an S-shaped curve. The same disinfectant, if you increase the concentration multiple folds, so definitely that concentration always it affects this disinfectant activity. So definitely you will get a graph C. So where initially there will be a very fast killing effect, and then later on, because of some resistant organisms, that will be slowed down slowly. Nearly it will be slowed down. So this is how you will get the death curve is obtained in the disinfection process, when you plot a graph of the time versus log of percentage of the survivors. But the matter of fact is that this survivor time or the survivor curve, it is not constant. So we can't say that every time we'll get this type of the curves. So this is influenced by a number of factors, especially the concentration of the disinfectant. So many scientists, many microbiologists, they have studied this. So the main conclusion of these workers was that the survival time curve is not constant, and its shape is being influenced by the number of factors, especially the concentration of the disinfectant which is being used. So here only you have seen that when you increase the concentration, so you got this, instead of the sigmoid curve, you got this C curve. So here, if you take the example of the, we'll take an example of the same graph. When you compare it with the different concentration of the phenol. Initially, if you plot a graph of concentration of the phenol at a concentration of 0.66 percentage weight by volume concentration, so this is the graph what you are going to get. So that means the time taken to kill all the organisms will be very less. You can see the time taken here is less. It will be almost near 20 minutes. But when you plot a graph with the concentration by reducing the concentration, that is 0.55 percentage weight by volume, so the same time it will be increased. The time to kill all the organisms will be increased. It will be more than 30 minutes. And when you, same thing, if you further reduce just slightly by 0.05 percentage, so when you're doing this experiment with 0.5 percentage weight by volume of the phenol, so this is the graph. And here the time taken to kill all the organisms will be more than 50 minutes. And then further reducing the concentration, I think we have lost the disinfectant activity of the phenol. So we are not going to achieve the complete destruction of the organism. Only few organisms will be killed, and the remaining resistant organisms will not be killed here. So we are not going to reach to the zero level here with this 0.44 percentage. So this is the effect of concentration. So anyway, we are going to study this in detail when we study this the effect of concentration, that is on the disinfectant activity. So that means overall the survivor time curve is not constant. So it depends on the concentration and many other factors. But usually what we deduce is that it's a first order kinetics. So naturally, theoretically, it should follow the first order kinetics, that is K is equal to 1 by T log P by B. And when you normally what you get is a sigmoid curve, and when you increase the concentration, we get a less than linear curve, so that is a C-type curve here. So that is the effect of time on the disinfectant activity. One second. So second factor, which is another important factor in the which will affect the disinfectant activity, is the concentration. I have already told you. So how this death curve, it changes with the change in the concentration. So the relationship between the concentration and the time taken to kill the organisms at a given temperature, it is exponential in nature. So that means, so here the three things will come into the picture, three different factors. One is the concentration, time, and the temperature. So we are going to discuss the temperature in the later stage. So here the second one is the concentration. So the relationship between the concentration and time taken to kill the organism at a particular temperature is always, it's an exponential in nature. That means doubling the concentration is considerably, it will be more than the half the rate if the concentration exponent is greater than the 1. So what is this concentration exponent? So if the concentration exponent is more